In the first three lectures of this week's lecture series, we've considered the idea of haplotypes. In other words, our genome is made up of chromosomes, and along those chromosomes are points of meiotic recombination within a population. These points of meiotic recombination lead to the exchange of alleles between homologous chromosomes. Alleles that are very close together tend not to be recombined by meiotic recombination and so occur together in a population. Alleles that tend to be seen to occur together in a population are called haplotypes. It reflects a property of these alleles called linkage disequilibrium, which indicates that they are very, very close to each other on the same chromosome. Haplotypes are very important in mapping the genetic basis of human disease, and we will return to this idea in future lectures. So we've talked a lot about genetic variants, single nucleotide polymorphisms, but really where do they come from? How do mutations occur in a population? What this graph shows here is a number of genetic variants across the genomes in different populations. So these are counts per individual that we see here. And you can see that quite clearly they're highest in Africans. In this case, they're African American, and they tend to be lower in Europeans. They have now been categorized into different types of mutations. Synonymous, which is where an amino acid is left unchanged. In other words, these are changes that occur within genes, or rather the coding sequence of genes. Missense, this is a change in amino acid. Deleterious types of variants, these are ones that have negative outcome for health. Unique, so we each carry 23 to 30 unique variants. Splice site, this affects the way that RNA is spliced. And nonsense, in which an amino acid is substituted for a stop codon, which is a very deleterious type of mutation. This figure here gives the rough breakdown of where we find polymorphisms in the human genome. Genes are made up of introns, which is non-coding DNA, and exons, which code for proteins. If you remember, only a very small part of our genome, about 1%, includes protein-coding genes. Protein-coding genes, in turn, are made up largely of exons, are made up largely of introns, with exons often being a smaller component of the gene. So it's not surprising then that most of the polymorphisms that we see occur in introns. We see a much lower number of polymorphisms in the promoter, about 4%, enhancers, about 8%. Now, these difference in numbers reflect the fact there's a lot more introns, so the likelihood you will see a polymorphism in an intron rather than an enhancer or a promoter is higher. But it also reflects the functional role of these different parts of our genome. Promoters and enhancers are important in gene regulation. Any change which will detrimentally affect the way a gene works will not be selected for. So that also selection maintains a lower rate of substitution in a functional element than it does in a region that is better able to tolerate change. We see that missense mutations, an alanine to a serine, for example, you can see a single base pair change, just the G to the T, about 0.51%, so much lower than promoters and enhancers too. Silent mutations, in which an amino acid stays the same, about 0.57%. So if you look at the relative occurrence of polymorphisms, SNPs, across a gene, introns by far and away have the highest number, followed by enhancers, promoters, and finally the exons themselves. This is what we call a frameshift mutation. So let's have a look at this. You can see here In the top line, we have the coding of the triplets for an amino acid. For example, ATG encodes the amino acid methionine, TCC is serine, and so on. Now, by inserting just one base pair, for example, here, what happens is that it changes the reading frame. In other words, you now make a different type of protein. So let's look here. We have a TAC-ATG leading to a tyrosine methionine, whereas the insertion of a single T base pair, or rather, excuse me, the deletion of an A base pair maintains the tyrosine, but now we have a cysteine followed by a glutamate, leucine, leucine, and so on, until we see a premature stop codon. So a frame shift mutation, and here we have a 2 base pair deletion, 1 base pair insertion, 2 base pair insertion. A frame shift mutation is an insertion or deletion of greater than or less than 3 base pairs. It changes the way a sequence is read, leading to, at the point of the insertion or deletion, a change in the amino acids and the introduction at some point of a premature stop codon. They are a highly deleterious mutation and are most often seen in cancer. Mutations can be inherited or familial, or they can be sporadic. Most cancers are due to sporadic mutations. A child is born with an autosomal dominant disorder, for example, and there is no family history. How could that occur? So a rare new mutation can arise in the germline of one of the parents through an error in meiosis, for example. Germline mosaicism, this means that the parents effectively have 2 genetic makeups. That can be an error during mitotic proliferation of the germ cell. The mutation, of course, could also occur in the child. What we see here is the development of the female and male gametes. During early fetal development, in both cases, there is mitotic proliferation. And in the males, that is followed by mitotic arrest. The females, however, enter meiotic arrest after the mitotic proliferation. Their final meiosis is not completed until the maturation of an egg during and after puberty. Males, on the other hand, undergo mitotic proliferation and enter meiosis during and after puberty, as we see here. This difference in the development of the gametes is actually one of the reasons for the increase in aneuploid disorders, such as trisomy 21, as the age of the mother increases. And this is correlated with the length of time in which the egg has been held in meiotic arrest. So let's see what we have here. Here is the ovary. There is the primordial germ cell that we see here, and in the testes as well. So there are repeated divisions, mitotic, and early mitotic proliferation. If a mutation occurs in one of these cells during early mitotic proliferation, then that mutation will spread through anywhere up to 50% of all eggs or sperm in that parent. If, however, the mutation occurs during meiosis, then the number of eggs or sperm that carry that mutation would be much lower. So depending on where the mutation occurs, increases or changes the risk of the child of inheriting that mutation. If it has occurred during mitotic proliferation, there's a much higher number of eggs or sperm that carry the mutation, where if it's in the meiotic phase, then the number of eggs or sperm can be very, very low. So how likely is it then that a gene will carry a mutation? Duchenne muscular dystrophy and Becker muscular dystrophy are X-linked diseases. Duchenne is the more severe clinical phenotype, diagnosis around 5 years of age, cardiac muscle failure in about 15% of patients, and the median age of survival is about 17 years. So a very severe clinical phenotype. Becker muscular dystrophy, which is this muscle weakness, is similar to Duchenne, but has a more benign phenotype. Age of onset around 12 years and a life expectancy of 40 to 50 years. The dystrophin gene, which is the gene that carries a mutation that causes one of the other of these dystrophies, is huge. It covers over 2 million base pairs. It has 79 exons, and 93% of this gene is introns. So a huge gene, a complex gene. And the size of the gene means it is more often the risk of carrying a mutation is increased. There are a number of different mutations which are associated with these dystrophies. So these result in changes to specific exons. Now, what happens is that there can be a deletion of exons 43, 4, 5, or 6 that will result in a frame shift. And we'll see why that is in a minute. However, if the deletion of an exon 41, 42, or 47 occurs, it will not result in a frame shift mutation. A frame shift mutation, as you remember, results in a change in amino acid sequence and the introduction of a premature stop codon. And this gives the more severe Duchenne muscular dystrophy phenotype. And here is the dystrophin protein. You can see it just here. It links the actin cytoskeleton with the extracellular matrix and so provides support and stability for the muscle. Key to understanding this difference in the phenotypes between Duchenne and Becker muscular dystrophy is the structure of the exons. Some of the exons are made up of a complete number of triplet codons. For example, exon 1 and exon 2 both contain complete numbers of triplet codons. When they are spliced together, they will form a reading frame that's I, M, C, A, D, and so on. However, there are other exons in the dystrophin gene which are not made up of complete numbers of triplet codons. For example, exon 1 in this case, ends with 2 base pairs. The following exon begins with a single base pair. The splicing together of exon 1 and 2 is required to generate this triplet codon sequence here. Deletion of either of these exons will, of course, result in a frame shift mutation and the more severe Becker muscular dystrophy. So this is a very large gene. It's a large gene which has a clinically significant number of deletions in exons. These exons are made up of either complete number of triplet codons or incomplete numbers of triplet codons. Depending on which type of exon is deleted, will result in either Becker or Duchenne muscular dystrophy. We've also talked about tandem repeats. These are the microsatellites. We've talked about these in the context of Huntington's disease. During replication, these repeats can be misaligned, leading to looping out, for example, and the expansion of these triplet repeats. This can happen as part of an error in DNA replication and have no health consequences for the individual. Or it can be pathogenic, where there are many, many repeats in a gene and we end up with a disease such as Huntington's disease. And we've already talked about this, where the increasing number of repeats through the generations will lead to an expansion of the CAG trinucleotide and the onset of Huntington's disease, something we call genetic anticipation. So single nucleotide polymorphisms, unlike the complex tandem repeats, are just single base pair differences. They have 2 alleles, for example, an A and a G allele, and they are usually common in a population, making up at least 1% of the minor allele will be a minimum of at least 1% in a population. But all of these alleles, of course, arose from rare mutations.